Bone cells. Bone is a living, dynamic tissue in which the processes of formation and breakdown are constantly taking place. To control this continuous remodeling, the body has four specialized bone cells: osteoprogenitor cells, osteoblasts, osteocytes, and osteoclasts. The coordination among these four cells is what maintains bone strength, flexibility, repair, and mineral balance. Let's start by talking about osteoprogenitor cells. These are the early undifferentiated cells that give rise to osteoblasts, the bone-forming cells. Osteoprogenitor cells are derived from mesenchymal stem cells and are considered the raw material for bone formation. These are immature precursor cells which later differentiate into either osteoblasts or chondroblasts. Structurally, these cells are very simple and spindle-shaped, much like fibroblasts. Their nucleus is elongated, oval-shaped, and located at the center of the cell. These cells mostly lack cytoplasmic organelles because their main role is just proliferation and differentiation, not heavy matrix synthesis. These cells are present in the inner layer of the periosteum and endosteum, from where they get activated to form new bone. Functions. Whenever the body needs new bone during fracture healing or growth, osteoprogenitor cells convert into osteoblasts, especially when they receive a good supply of oxygen. Oxygen acts as a stimulus for osteoprogenitor cells. In the area where these cells are present, if there is a lack of oxygen, then these cells mostly convert into chondroblasts, which are the cells that form cartilage. This means that a low oxygen environment pushes osteoprogenitor cells in the direction of forming cartilage. Cartilage is generally found in areas where blood supply is low, so low oxygen naturally supports this process. On the other hand, if there is more oxygen in this area, osteoprogenitor cells convert into osteoblasts, which are the cells that form bone. A high oxygen environment is ideal for bone formation, so increased oxygen activates and supports osteoblasts. When osteoblast progenitor cells mature and become active, they turn into osteoblasts. Osteoblasts are active bone-forming cells. These cells produce the bone matrix. They are the cells that form bone. In other words, they are the factories of bone that create new bone matrix. Its structure is completely different. It is typically cuboidal or columnar in shape, especially when they are active and forming bone. It has a round-shaped nucleus in the center. Due to the abundance of ribosomes, its cytoplasm is basophilic. Basophilic tissues or cells strongly absorb basic, that is, alkaline dyes. They appear blue or purple under the microscope. Inside, the rough endoplasmic reticulum and Golgi apparatus are well developed because these cells produce and secrete collagen type I and other bone proteins. They are arranged in a single layer on the outer and inner surfaces of the bone and perform four major functions. In the stage of formation, osteoblasts create the organic matrix known as osteoid, which is the structural unit of compact bone, and it is composed of collagen type I, glycosaminoglycans, and glycoproteins. After forming the osteoid, the osteoblast itself mineralizes this matrix. For this, it releases enzymes like alkaline phosphatase. This stores calcium phosphate crystals and converts the osteoid into hard bone. Regulation of bone remodeling. Bone remodeling is a continuous process in which old bone is broken down and new bone is formed, so that the bone remains strong, healthy, and updated. Osteoblasts produce two important proteins that keep the bone remodeling system in balance. The first is rankle. This protein is released by osteoblasts and stimulates osteoclasts. As a result, osteoclasts become active and start the resorption, that is, the breakdown of bone. The second is osteoprotegerin. It is the natural blocker of Rank-L. Osteoprotegerin inhibits osteoclasts. That means it reduces their activity so that bone resorption does not become excessive. In other words, osteoblasts not only build bone but also control the mechanism of bone breakdown. Hormone response 19 stimulates the Rankle system while osteoprotegerin counteracts it. Osteocytes are mature bone cells found in lacunae that maintain bone tissues and regulate bone remodeling through their long canalicular processes. These are the most abundant cells found in bone. That is, throughout the bone tissue, these cells are present in the greatest numbers. Osteocytes are the mature form of osteoblasts, but they are very different from osteoblasts in both structure and function. Osteocytes reside inside small cavities within the bone, which are called lacunae. Their structure is spider-shaped, with many long and thin cytoplasmic processes. These processes extend through microscopic channels called canaliculi, which directly connect each osteocyte to other osteocytes and to osteoblasts present on the bone surface. At the end of each process, there is a gap junction that allows ions, nutrients, and chemical signals to be transferred from one cell to another. Functions. Osteocytes perform these functions. Mechanosensation means sensing mechanical stress, that is, pressure or force. Osteocytes are the sensors of the bone. When we walk or lift weights, the pressure that is exerted on the bone is sensed by the osteocyte through its long processes. If there is more or less pressure on any part, it immediately senses this change. Intercellular communication. Each osteocyte lives in a small space called a lacuna. Many fine pathways emerge from this lacunae, which are called canaliculi, and they spread out in all directions. Through these canaliculi, osteocytes are connected to each other and to the osteoblasts present on the surface. They share nutrients, waste, calcium signals, and regulatory messages with each other. In other words, the entire network forms the bone's own communication system. Regulation of bone remodeling signals. Bones are constantly breaking down and rebuilding, a process known as bone remodeling. Osteocytes are the main controllers of this entire process. If they sense even a slight decrease in pressure, they signal the osteoclasts to start resorbing bone, and when they sense increased mechanical load, they activate the osteoblasts to start forming new bone. Control of osteoblast and osteoclast activity. Osteocytes release certain specific molecules, that is, chemical messengers, which directly control the activity of osteoblasts and osteoclasts. One important molecule among these is sclerostin, which inhibits osteoblasts and slows down bone formation, and when it is released less, the opposite process occurs. Similarly, when bone resorption is needed, they produce molecules like rankle to activate osteoclasts. Osteocytes act like traffic controllers, deciding when bone should be formed and when it should be broken down. Maintain mineral homeostasis in bone matrix. Osteocytes maintain the balance of minerals within the bone. They regulate calcium phosphate and other ions between the bone matrix and the bloodstream. When the body needs calcium, such as for muscle contraction or nerve signaling, osteocytes slightly dissolve the surrounding bone to release calcium. This process is called osteocytic osteolysis. An osteoclast is a large multinucleated cell that breaks down and resorbs bone during the process of bone remodeling. Osteoclasts are the cells that resorb or break down our bone and remodel it. Just as osteoblasts form new bone, osteoclasts dissolve old, weak, or damaged bone. This process is half of bone remodeling. On one hand, bone is formed, on the other hand, it is broken down and replaced. Structure of osteoclast. Understanding its structure is very important because its design is specifically made for bone resorption. Multinucleated giant cells. An osteoclast is a very large cell that typically contains anywhere from 3 to 20 nuclei. The advantage of having more nuclei is that the cell's metabolic capacity becomes very high, which allows bone resorption to occur very rapidly. Ruffled border. This is the most unique part of these cells. This border is a wave-like folded surface from where the osteoclast releases acid and enzymes onto the bone. More force means more activity, that is, faster resorption. Sealing zone. The osteoclast attaches its base to the bone surface, creating a strong sealed area. It is within this sealed zone that the broken down bone matrix accumulates. This small isolated zone is called the resorption lacuna or Hauschup's lacuna. Inside these cells, there are many acidic vesicles and lysosomes that contain enzymes like cathepsin K and tartrate-resistant acid phosphatase, or TRAP for short, as well as acid hydrolases. These enzymes break down bone. Origin of osteoclast. Unlike osteocytes, osteoclasts do not come from the osteoblast family, but rather from the monocyte macrophage lineage, meaning they are actually derived from immune system cells. In the bone marrow, monocyte precursors fuse together and then form a giant multinucleated osteoclast. Location of osteoclast. These cells are usually found where bone breakdown is taking place. On the surface of the endosteum, at the edges of trabeculae, inside Haversian lacunae, and at remodeling units where osteoblasts and osteoclasts work together. Functions of osteoclast. Their basic function is to break down bone, but they perform this task in a very scientifically controlled manner. Bone resorption. Osteoclasts release acids which dissolve the mineral part of the bone, that is, hydroxyapatite. After this, they also digest the fibers and organic matrix through enzymes. Maintains calcium level. When bone is dissolved, calcium and phosphate are released into the blood. In this way, osteoclasts regulate systemic calcium levels, helping in bone remodeling. Osteoclasts remove old bone, after which osteoblasts form new, fresh bone, and this cycle continues throughout life. How they dissolve bone. Osteoclasts perform these two functions for bone resorption through their ruffled border. Acid secretion. Hydrogen ions are released onto the bone surface by the ATPase pump, and these acids dissolve the mineral surface of the bone. Enzymes release. After the minerals are dissolved, catepsin K enzymes break down the fibers, and protease enzymes digest the matrix. All these breakdown products are then sent into the blood. Regulation of osteoclast activity. This activity is maintained due to two things, rankel and osteoprotegerine. Osteoblasts release rankel, which stimulates the rank receptor present on osteoclasts. Once activated, the osteoclast starts bone resorption. Osteoprotegerine is also released by osteoblasts, and by blocking rankel, it stops osteoclast activity, that is bone resorption,